Today we will see the C4 cycle or HSK pathway. Up to 1965, it was believed that the CO2 fixation during the photosynthesis of higher and lower plant takes place only by Kelvin cycle. But the Koschak, Hurt and Burr in 1965 demonstrated that in a sugarcane leaves, first stable compound is a 4 carbon rather than 3 carbon as in a Kelvin cycle. By using C14 carbon dioxide, this was confirmed by Hatch and Slack in 1966 and shown that the first stable compound is oxaloacetic acid. Hence, they proposed a new alternative path of carbon in a sugar cane. To honor them, it is known as HSK cycle, and due to 4 carbon first stable compound, it is also called as a C4 cycle. Later on, these observations have been confirmed in other monocots like maize, jawar, bajra, cypress, etc., and some dicots like amaranthus, euphorbia, atriplex. These plants are thus known as C4 plants. They exhibit special anatomical features in the leaves such as a kranz anatomy. In this leaf anatomy, C4 plant possesses two types of chloroplast. These are the mesophyll and bundle sheath chloroplasts. The reactions are divided into three parts. That is the reactions in a mesophyll chloroplast, reactions in a bundle sheath chloroplast, and a regeneration of PEP, that is the phosphoenol pyruvate in a mesophyll chloroplast. Characteristic of C4 plants. The first character, that is the leaves of C4 plants, possesses a kranz anatomy. That means there is a presence of two types of the chloroplast, that is the mesophyll cell chloroplast and bundle sheath cell chloroplast. Second one, that is the chloroplast in a C4 plants are dimorphic. That means they possesses two types of the chloroplast. The mesophyll chloroplasts are smaller in size, randomly arranged, many in number, faint green in color, and follows HSK cycle. They are a granule chloroplast, while the bundle sheath chloroplasts are larger in size, rarely or centripetally arranged, few in number, dark green in color, and follow the Kelvin cycle. Hence, they contain the starch grains. They are agranule chloroplasts. A third important characteristic, that is the PEP carboxylase, is present in a mesophyll chloroplast, while the RUDP carboxylase is present in a bundle sheath chloroplasts. The fourth important characteristic, that is the first stable compound in them, is the oxaloacetic acid, that is the 4 carbon compound structure. We will see first of all the reactions in a mesophyll chloroplast. The first reaction, that is the primary CO2 acceptor of this cycle, is a phosphoenol pyruvic acid, that is the PEP, present in a mesophyll chloroplast. It accepts atmospheric CO2 in presence of phosphoenol carboxylase and water to form a first stable 4 carbon compound structure, that is the oxaloacetic acid, that is the OAA, along with a phosphoric acid. Well, we will see the reaction, that is the PEP plus carbon dioxide plus water molecule in presence of enzyme PEP carboxylase. There is a synthesis of OAA, that is the oxaloacetic acid, and phosphoric acid. In a second reaction, that OAA is quite unstable and is converted into either malic acid in a most of C4 plants or aspartic acid in a few C4 plants. The OAA is reduced to malic acid by using light generated NADPH2 in presence of enzyme malic dehydrogenase. Well, we will see the reaction, that is the OAA, oxaloacetic acid. It interacts with NADPH2. That reaction, which is the reversible reaction, there is a synthesis of malic acid and NADP plus. In a few C4 plants, that oxaloacetic acid is converted into a aspartic acid in presence of enzyme aspartic transaminase. We will see the reaction, that is the OAA, in presence of enzyme aspartic transaminase. There is a synthesis of aspartic acid. That reaction, which is the reversible reaction, this malic acid or aspartic acid is then transported to a bundle with chloroplasts. Reactions in a bundle with chloroplasts. The first reaction in a bundle with chloroplast, that is the malic acid, is a decarboxylated to produce a pyruvic acid, that is the 3 carbon compound structure, and a carbon dioxide in presence of enzyme malic. In the same reaction, a one molecule of NADP plus is reduced to a NADPH2. We will see the reaction. In a reaction, that malic acid, it interacts with that NADP plus. There is a formation of pyruvic acid, that is the 3 carbon compound structure, synthesis of NADPH2, and there is evolution of CO2. In a second reaction, particularly in bundles with cells, both CO2 and NADPH2 produced in this reaction enters into a Kelvin cycle. The CO2 is accepted by a 5 carbon compound structure, that is the ribulose 1,5 diphosphate. We can say that is the RUDP, to yield or gives a 2 molecules of a 3 phosphoglyceric acid, that is the 3-PGA. We will see the reaction, that is the CO2 plus RUDP. There is a synthesis of 2 molecules of 3-PGA. This PGA is later used for the synthesis of carbohydrates and also for the regeneration of RUDP as usual. When aspartic acid comes in a bundle with cell chloroplast, it is the first converted to OAA, which is then decarboxylated to carbon dioxide and pyruvic acid. We will see the reaction, that is the aspartic acid, it is converted into OAA, then there is a synthesis of pyruvic acid and there is evolution of CO2. The third reaction in HSK pathway or C4 cycle, that is the regeneration of phosphoenol pyruvate. This pyruvic acid is transported back to the mesophyll chloroplast, where it is phosphorylated with one ATP molecule of light reactions to produce a phosphoenol pyruvic acid in presence of enzyme pyruvate phosphate dikinase. We will see the reaction, that is the pyruvic acid plus ATP in presence of enzyme pyruvate phosphate dikinase. There is a synthesis of phosphoenol pyruvate and ADP. After completion of mechanism, we will see that is the significance of HSK pathway or C4 cycle. The first significance, that is the C4 plants are more efficient in photosynthesis and CO2 fixation, which is a very fast one. Second one, that is the low concentration of carbon dioxide or high concentration of O2 does not affect the rate of photosynthesis in a C4 plants. A third significance, that is the high temperature also does not affect the C4 cycle. The fourth significance, that is the photorespiration is absent in a C4 plants. And a last significance, that is the H and Slack reported that the C4 plants thus are well adapted to grow under a low water content and high temperature. Thank you.